So today, as my title indicates, I'm going to talk to you about the mechanism that regulated the different tumor states and their implication for cancer therapy. So as you know, and you heard many times in this meeting, within a given tumor, not all tumor cells are alike. Some cells proliferate, other cells differentiate, some cells invade the underlying tissue, circulate in the blood vessels, and can seed to distant organs and can lead to metastasis. And in addition, when you give treatment, not all cancer cells respond identically. Some cells, undergo differentiation, some cells die, or some cells escape therapy and induce tumor relapse. So it's very important to understand the basic mechanism that regulated tumor heterogeneity in these different circumstances. So I'm going to talk to you today about one single cancer, which is the squamous cell carcinoma. And I'm going to, my talk will be focused on the skin squamous cell carcinoma, but I believe that, other type of squamous cell carcinoma may follow the same paradigm. And so the squamous cell carcinoma is the second most frequent skin cancer. It affects about 1 million of patients per year throughout the world. And in most of the cases, it can be cured by surgical excision. But nevertheless, in 5 remaining percent, it can lead to metastasis and then be associated with a much poorer prognosis. But much more importantly, squamous cell carcinoma is the leading cancer in oral cavity, in head and neck, in the esophagus, and in a proportion of non-small cell lung carcinoma, which is associated with much higher mortality. And for this reason, it's important to really understand the mechanism that leads to tumor heterogeneity in squamous cell carcinoma. So in the model that I'm going to use, and in skin squamous cell carcinoma, it's RAS and p53 pathway that are frequently mutated, but I will show you, there's other genes that are mutated as well, and for which I'm going to talk to you a little bit about. So the first question that we had about the tumor heterogeneity was, do the cell of origin of the cancer can control tumor heterogeneity? And for that, we rely on lineage tracing experiment in which we put the same oncogene, KRAS G12D mutation and p53 loss of function, together with YFP expression to follow the cells that we targeted. And we target two distinct compartments of the skin epidermis, the interfollicular epidermis and the hair follicle stem cell and their progeny. And what we found is that the same oncogene leads, in these two different cell types, leads to a very distinct tumor phenotype. The one that targets the interfollicular epidermis gives rise to what we call a well-differentiated squamous cell carcinoma. Any pathologist in the world would see this image, H&E pictures, would say, well, it's called squamous cell carcinoma because you have these keratin pearls, and as you see here, it's very relatively homogeneous. They all express keratin 14, which is an epithelial marker, and none of the cells express vimentin, a mesenchymal marker. But in the hair follicle-derived tumor, we observe a most frequent type of tumor, which what we call mixed CC, which was a tumor in which there was an epithelial part of the tumor, but also some cells that undergo EMT lost the epithelial expression, marker expression, and began to express a marker of the mesenchymal thing, so a classical example of EMT. And so we could track down very easily EMT using EpCAM expression, which is a cell surface protein, an adhesion molecule, a little bit like a E-cadherin. And you see here there's a beautiful bimodal expression of EpCAM in mixed SCC that undergo EMT, and there's even mesenchymal-like tumor which are completely EpCAM negative. And so, as you can see here, analyzing several hundreds of those tumors, we can see that they almost all invariably undergo EMT with certain degree. Some cells, there's only a few percentage of the cells that undergo EMT, and the cells, most of the tumor cells had a mixed phenotype, and some cells that completely mesenchymal. So what I show you here, that tumor heterogeneity can be dictated by the cellular origin of cancer, which was, a very important finding, I believe. And that is due to the fact that the oncogene that can co-opt, it can co-opt the transcriptional and epigenetic machinery that are present in the cell of origin and rewire these cells into distinct differentiation program, and the hair follicle will give rise to this EMT phenotype. So, why this is important, and I will illustrate that by several examples. The first one is that EMT in those tumor models is associated with a high prevalence or high incidence of metastasis. And so when you check this hair follicle-derived tumor, there, about 30% of those present lung mets, and about 50% of those expressing lymph node metastasis. In sharp contrast, when you check at the metastasis in the interfollicular-derived tumor, there was a very low frequency of metastasis. And in each mice that present metastasis, there was at least one tumor that present some kind of EMT at least in one of the tumors present in this mice, suggesting that in this mouse model, metastasis is almost invariably associated with EMT. So the reason why people were skeptical about EMT and pathologists in particular were the fact that when you look at the metastatic cells in the distant organ, there were very little sign of EMT. But in this model, this is exactly what we found as well. Whereas the primary tumor here were really mesenchymal, the metastases were really epithelial, and at least 70% of the lungs metastases do not present any sign of EMT, and in most of the cases, they revert to a completely epithelial tumor phenotype, suggesting that the reversion from EMT to MET to the epithelial state is important for metastatic outgrowth. So, for a long time, it's been so that, EMT was a binary process, and when you check this FACS plot of EpCAM expression, is indeed consistent with this idea. As you see here, there's a population that is purely EpCAM positive and population that are purely EpCAM negative, and you don't have, a big continuum of cells in between the two, suggesting that at least some of the state here occur through a binary decision. But something that we could not exclude, that being hidden in this single peak of EpCAM expression or not EpCAM at all, there's an heterogeneity of tumor state that can account for a diversity of EMT tumor state. And that's what we tried to assess by using a very neat approach, I think, because we screened this primary tumor that present EMT with a bunch of cell surface marker and check whether these markers were either homogeneously expressed, which suggests that the population is homogeneous, not expressed, we cannot conclude anything, or heterogeneously expressed. That would indicate that the population is indeed heterogeneous, and more things are needed to understand their functional diversity. So when we did that, and screened these 200 cell surface marker, we found that the EpCAM positive tumor population were really homogeneous. Almost all the markers were homogeneously expressed. In sharp contrast, when we checked the marker expression in the EpCAM negative tumor fraction, the marker expression were very heterogeneous. More than 50% of the markers were heterogeneously expressed, and very often in a bimodal pattern of expression. So that was very interesting. Then, we combine those markers 2 by 3, 2 by 2, 3 by 3, until we could resolve kind of nicely the different heterogeneity. And we found that 3 cell surface markers were sufficient to deconvolute the heterogeneity we found in this EpCAM negative population. And very interestingly, those markers were not completely random when we realized what they were. They were CD51 and CD61, which was alpha V beta 3 integrin, and for which the expression were previously reported to be heterogeneously expressed in certain cancer stem cells, such as the breast, by David Chires' group in San Diego. And CD106 was VCAM1, that was at that time recently identified by the group of Jan Massagé as a marker of metastatic initiating cells in solid tumors. So, we were quite happy with that, and so we checked, what would be the type of EMT population that we could find in this different cellular fraction the triple negative, so the one that do not express any of the three markers, CD106, CD51, and the double positive, and the triple positive. And for that, we isolate this population by fact, cytospin them, and stain them with K14 and vimentin to assess their epithelial and mesenchymal characteristics. And what we found is as soon as you lost EpCAM expression, you begin to express vimentin. But at the beginning, this vimentin expression is also accompanied by the sustained expression of a epithelial marker. And that's what we call hybrid EMT tumor cells. And this terminology of hybrid tumor cells can be called by some people partial EMT or quasi-mesenchymal EMT. All this terminology refer to the same thing, which is the co-expression of an epithelial and a mesenchymal marker in the same cells. So when we test the metastatic potential of these different fractions, here's where the big surprise arises. The most metastatic cells were not at all the most mesenchymal population, the triple positive population, but were the triple negative and the CD106 population, the most early hybrid population that we found in this tumor. And so that's indicated that a dual expression program of EMT and epithelial state is important for metastatic colonization and seeding. And very importantly, since the publication of this paper, there's been a dozen and dozen of papers that has been published thereafter showing that these hybrid populations were truly the most metastatic population in many different tumors, even involving the most cutting-edge methods, such as the lineage tracing strategy that has been done by certain investigators, and that has been shown in many different cancers. In addition, this triple hybrid population were also the cells that present the most circulating tumor cells that we found in the blood of these mice presenting EMT tumor cells. So I didn't have the time to present you all the functional data here, but what I show you is that EMT is not a binary process. It's accompanied by distinct transitional state. It's very much a continuum of tumor state. There are many plasticity in between these tumor states. Nevertheless, some tumor states are more stable than the other, and in particular, the early hybrid population and the late EMT population. The epithelial are the most proliferative. The early hybrid and the hybrid EMT state are the most metastatic. And the late hybrid and the late mesenchymal tumor cells, the pure EMT tumor cells, are the more invasive, but are not the more metastatic. So, and also we looked in this paper, where these tumor cells were located, and we found that they were located in different niches, and so we thought, it was mainly regulated by an epigenetic and a transcriptional program that can be itself regulated by the tumor microenvironment. But we didn't know whether there were mutations that can confer this state to the tumor. And that's what, we subsequently found is FAT1, which is by far one of the most mutated tumor suppressor gene in squamous cell carcinoma, and is the second most frequently mutated gene in head and neck, second most frequently mutated gene in oral carcinoma, second most frequently mutated gene, or within the 5 most frequently mutated gene in non-small cell squamous cell carcinoma. And when you deleted FAT1, it activates calmodulin kinase, which in turn activates SERCA-NIAP kinase, and which activated 2 distinct differentiation programs, the EMT program and the epithelial program. And by identifying what was downstream of FAT1, we identified tumor vulnerability in those tumors. And when we treated mice harboring FAT1 mutation with SRC inhibitor, they were way much more sensitive to the SRC inhibitor than the wild-type counterpart, opening the way of more precision medicine for patients that present FAT1 mutation. So now I'm going to switch gears and kind of showing you that all this knowledge can be used to try to target this EMT state and potentially be used in clinical trial, and also trying to identify what could be the molecular mechanisms that are regulated in EMT tumor cells that made them resistant to chemotherapy. And so when we profile these EMT tumor cells and all these different fractions, we look for molecules for which there were drugs tested in clinical trials and could be available to try to target the EMT tumor state. And we found that natrin-1, which is a guidance molecule, were highly upregulated in EMT tumor cells as soon as the cells undergo this early hybrid EMT transition. And so I knew that Patrick Mellen and Agnès Bernet, two researchers in Lyon, had developed a monoclonal antibody that they were testing in clinical trials in patients with cancer to try to improve their response to chemotherapy. And so we team up with the group of Patrick Mellen, and Justine Langrand came from Lyon and finished his PhD in my laboratory trying to understand the role of anti-natrin-1 in regulating EMT. So what I show you here, natrin is upregulated in EMT tumor cells. And very interestingly, when you give this anti-natrin-1 monoclonal antibody to the mouse that has been induced and that will develop EMT tumors, they present a strong decrease in tumorigenesis, but also a major decrease in the proportion of EMT tumor cells. And we could show that when we combine one of the current treatment, 5-FU cisplatin that is given to patients with squamous cell carcinoma with this anti-natrin-1 antibody, we can sensitize the tumors to chemotherapy and prevent their tumor progression. So we try to understand, what could be the molecular mechanism behind the natrin-1 treatment here, and how it can impact EMT and sensitize the cells to the chemotherapy. So for that, we perform single-cell transcriptomic analysis, and so that's a typical example of how this single-cell analysis show. It's exactly the same kind of state that I show you with the monoclonal antibody. You have first the epithelial, pure epithelial state, then you have the early hybrid EMT, so it's a population that lost FCAM expression but still maintain KF14 expression. Then you have the late hybrid EMT population which keep expressing keratin 8, another epithelial marker, and vimentin expression. And then you lost completely all epithelial marker exclusively expressing the mesenchymal marker. Interestingly, when we treated the tumor cells with anti-natrin antibodies, we found that there was a very distinct tumor state that were identified by single-cell transcriptomic, and there was essentially an epithelial state, an early and late hybrid, but very little of late EMT state that we found. And when we perform lineage trajectory to this, we found, as Bob Weinberg recently reported, that there's two lineage trajectory during EMT, one that leads to the late hybrid EMT and another lineage trajectory that leads to the late pure EMT population. And in contrast, when you check the lineage trajectory that occur upon natrin-1 inhibition, you find that the late EMT trajectory is completely gone, and you have another epithelial, like a super epithelial state trajectory, and you conserve this hybrid EMT trajectory. Now I'm going to show you, just in the last part of my talk, how the EMT tumor cells can resist to chemotherapy. And so that work has been done by, Maud de Bonny, who was an MD PhD student, and she's a head and neck surgeon. And what she found is, as was reported in other circumstances, in other models, that if you give cisplatin, so cisplatin in this presentation is made by CDDP, it's kind of the medic's words for cisplatin, and 5-FU, the EpCAM positive tumor cells, they were really sensitive and they present much higher level of apoptosis following cisplatin than the EpCAM negative tumor cells, as you see here. But it was not only, they were not only resistant to cisplatin 5-FU, they were actually resistant to a wide range of chemotherapy and replicative stress agents such as topotecan or etoposide, in addition to the cisplatin 5-FU. So, to try to understand, why they were resistant to chemotherapy-induced cell death, we try to look in our RNA-seq data, what could be the gene that were involved in resistance to chemotherapy. And our attention has been caught by this small GTPase, which is called ROGE, and is a member of the CDC42 family of GTPase. And there was one paper that were published some 10 years ago who showed that ROGE would prevent apoptosis following cisplatin 5-FU in melanoma cell line through a p53-dependent mechanism. And so, although, we didn't think that in our case p53 was important because, we delete p53 in our model, so all cells EpCAM plus or EpCAM minus present p53 deletion, but nevertheless, it looks like an interesting candidate. So for that, we generate loss of function of Rho-G and Rho-Q, its little brother that almost share more than 90% of homology with Rho-G. And as you can see here, knocking down Rho-G massively increase the cell death in response to cisplatin 5-FU, whereas the Rho-Q doesn't affect cell survival at all. The gain of function of Rho-G, in contrast, can prevent cell death in normally sensitive cell line, the epithelial cell line, EpCAM positive cells. And so gain and loss of function show the opposite effect and show that Rho-G is important for promoting resistance to chemotherapy. So we did, that was with sgRNA, and that was in vitro, so we turned out to what we very often do in our lab is to use in vivo model with knockout strategy, and so we remove the two allele of Rho-G, we got this flux allele from collaborators, and then there was also a small decrease in tumorigenesis, but a major decrease both in vivo of the EpCAM negative cells here in response to cisplatin 5-FU, and in vitro it was even more striking. And as you see here, that's after 24 hours is the proportion of caspase-3. Here's the tumor progression over the 3 weeks of tumor growth in vivo, and as you see here, there's a major difference when Rho-G is mutated in those tumors. So we wanted to understand the molecular mechanism for that. So we first did RNA-seq, and there was really little insight from this RNA-seq data. So we decided to do proteomic analysis because we know that very often DNA damage response is mediated by ubiquitination or phosphorylation or protein abundance and stability. And so we did a proteomic screen, and we found that there was a group of proteins that are regulated in a Rho-G dependent manner. And interestingly, when you check what are those proteins, we found that there was a huge amount of protein that were regulated by Rho-G that are important in a protein on nascent DNA and upon replicative stress. And so indicating that Rho-G may indeed regulating the DNA damage response. So we check that by gamma H2X, which mark an repair DNA break. And as you see here, Rho-G wild-type cells, the EpCAM negative cells, are really present really little amount of H2X as compared to Rho-G knockout cells from the same phenotype. So clearly, the EMT tumor cells were resisting to DNA damage by promoting the repair of the DNA lesion induced by the chemotherapy. And so to understand why we team up with Juan Mendez, which is, one of the world specialists in the origin of replication at the CNRO. And what Juan and his postdoc did, they did to measure the rapidity of the fork replication and the amount of replication that occur in the different conditions, epithelial, mesenchymal, or Roger knockout mesenchymal. And we found that although all the cell state, they slow down the fork of replication rate, the Roger positive EMT tumor cells, they recruit dormant of origin of replication to repair the DNA faster. And finally, to really understand what could be how it's regulating this origin of replication, we did pull down of Roger and following by mass spec. And we identify among the genes that Roger regulated a bunch of actin regulator, and in particular importin 9, which was a very key regulator that regulates nuclear actin. And nuclear actin has been shown by different groups to be important to regulate the DNA repair in turn. So we decided to check whether the EMT tumor cells present more nuclear actin, and it was indeed the case, and this nuclear actin was even further increased following chemotherapy in a Roger-dependent manner. And when we inhibit actin polymerization by l'antranculine B, we can sensitize now the tumor cells to undergo cell death, the CMT tumor cells to undergo cell death following chemotherapy. And finally, we could show that this increase of resistance to chemotherapy was due to the lack of recruitment of new origin of replication in an actin-dependent manner, and that is accompanied by a decrease in DNA repair. So, with that, I thank, all the people that has done this work, and, I cited them all along, and Justine Langrand and Maud de Bonny were the last two fellows that are still remaining in my lab. Eugenia Pastuchenko, who is now assistant professor at University of Brussels. And I thank also all the funding that allow us to do all this research. And I thank you for your attention, and I will be very happy to answer your questions.